it's Miss Fish, and I'm here to talk to you today about meiosis. So, meiosis, what is it? It's a cell division to produce gametes, which is another way to say sex cells, which is another way to say sperm and egg cells. So, gametes are sperm and eggs. There are two divisions with the result of 4 gametes, or 4 sex cells, meiosis 1 and meiosis 2. It's really important that you write those Roman numerals next to each one because otherwise I don't know which one you're talking about. Why are there 2 divisions? Non-sex cells or somatic cells have a total of 46 chromosomes. During meiosis, the number has to be cut in half so that when a sperm fertilizes an egg, the fetus has 46 chromosomes. Go ahead and pause now if you need to finish this slide. So what are the 2 steps to meiosis? Well, meiosis is similar to mitosis. It has to go through the interphase before it begins dividing. Remember, interphase is that stage where DNA is copied. After interphase, it follows the following order. So during the first division, it would go through prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. During the second division, it does not go through the second division. It does not go through interphase. It is completely skipped because our DNA has already been copied. We don't need to copy it again. So directly after the cytokinesis from the first division, it goes through prophase 2, metaphase 2, anaphase 2, telophase 2, and then one more cytokinesis. Go ahead and pause if you need to finish writing this slide. Prophase 1. Chromosomes are condensed, the nucleus disappears, and homologous chromosomes pair up. This does not happen in mitosis. The prefix homo means the same. A lot of people refer to same-sex relationships. So if you're in a homosexual relationship, you are in a relationship with someone who is of the same gender as you are. So it's the same. So homologous chromosomes are chromosome pairs, one from each parent, that are similar in length, gene position, and centromere location. The genes are in the same spot, but they may carry different alleles or traits. So here's a picture of what prophase 1 might look like. So notice here that my 2 homologous chromosomes, my larger ones, are paired up, and my smaller homologous chromosomes are paired up. Our spindle fibers are going to attach to each of the chromosomes, and our nucleus will disappear. Metaphase 1. Homologous pairs line up at the center of the cell. There is no order in how they line up. This is the law of segregation. Basically, since there is no order, each trait has a chance of being passed on. Crossing over can also occur during this stage. Homologous pairs swap parts of their chromosomes, essentially mixing up the results. Crossing over leads to this law called the law of independent assortment. What it stands for is that the inheritance of genes do not depend on another. I have brown hair and green eyes. My dad has red hair and green eyes, and my mom has brown hair and blue eyes. So just because I inherited my dad's green eyes does not mean I inherited my dad's red eyes. My dad's red hair. Red eyes. That's funny. Go ahead and pause now if you need to finish writing the slide. Alright, so here is a picture of what metaphase would look like. Metaphase 1, our homologous pairs are lining up in the center of the cell. Anaphase 1, homologous chromosomes are pulled apart in opposite directions. Sister chromatids are still intact. Go ahead and pause if you need to. Here is a picture of anaphase 1, our sister chromosomes. Our homologous pairs are being pulled apart from each other. Telophase 1. Chromosomes arrive at either end of the cell and the nucleus reappears. The cell begins to go through cytokinesis, where the cytoplasm divides. Go ahead and pause if you need to. Here's a picture of what telophase 1 might look like. Our nucleus is reappearing, our chromosomes are on either side of the cell, and our cells are going to begin dividing. Cells are now haploids, meaning there are half the number of chromosomes. Each cell has 2 copies of each chromatid. Cell takes a rest and then goes on to meiosis 2. Prophase 2 resembles prophase 1. Nucleus disappears, and the chromosomes move toward the center of the cell. Spindle fibers attach to the center of each chromosome. Go ahead and pause if you need to. Here's a picture of prophase 1. Our nucleus is going to begin dissolving and disappearing again, and we have our chromosomes getting ready to line up in the center of the cell. In metaphase 2, chromosomes line up along the center of the cell. A sister chromatid is on each side. And I'll show you a picture in just a second. If you need to finish writing, please pause the video. In metaphase, this is what it looks like in both of our cells. Here's one cell and here's our second cell. Our chromosomes are lining up along the center, and this is called a sister chromatid. So one side of the sister chromatid faces each pole. So I have one side on this side and one side on this side. Same thing over here. Anaphase 2. Sister chromatids are pulled apart toward the centrioles. The sister chromatids are now called sister chromosomes. In humans, there are now 23 sister chromosomes in each of the 4 cells. Go ahead and pause if you need to finish writing. So this is what anaphase 2 looks like. My sister chromatids are being pulled apart in opposite directions to either side of the poles. Telophase 2. Chromosomes are on either side of the cell and the cell begins to divide. Cytokinesis occurs. Go ahead and pause if you need to. So here's telophase 2. Notice that we are ending with 4 different cells that are completely unique. None of them are the same, and they each have 2 chromosomes, which is half of what we started with. We started with 4. So the end result of meiosis 2. 4 new cells are made, each with 23 chromosomes. If you're using my guided notes for this section, you'll notice there's another blank after the 23 chromosomes. Please ignore that blank. Go ahead and scratch it out. I apologize. I don't know what I was talking about there. The 4 cells will turn into sperm for males or eggs for females. Go ahead and pause if you need to. And why is meiosis important? If meiosis did not occur, then we would have too many chromosomes in our cells. Each generation would double in the number of chromosomes. For example, the first generation of people, whoever you believe that was, would have the normal 46 chromosomes. But then their second generation of kids, so their kids, would have 92 chromosomes. And then their kids would have 194 chromosomes, and so on. So we need to go through meiosis, or we'll end up with way too many chromosomes in our cells. Well, I hope you guys have a good day. And if you have any questions, let me know before or after class. And I'll see you in class tomorrow. I hope you have a good day. Bye.